Welcome to this lecture on evolutionary biology. Today, we are going to explore one of the most fascinating phenomena in nature, known as ring species. If you have ever wondered how one species can gradually transform into another, or why the line between species is sometimes blurry, you are in the right place. We will build our understanding from the ground up, looking at real-world examples and the mathematics that explain them. Let us dive in. Now that we have introduced our topic, let us start with an intuitive analogy. Imagine a paint color strip that starts as pure red on the far left and slowly fades into pure blue on the far right. If you look at any two adjacent shades on this strip, they look almost identical. Because the change is so gradual, every color blends perfectly with the one right next to it. However, if you take the far left red and the far right blue and put them right next to each other, they are completely different colors that do not match at all. Now, imagine this exact same concept happening with living creatures, spreading around a giant physical obstacle, like a massive mountain range. This beautiful, continuous gradient of life is what we call a ring species. Building on that color strip analogy, let us look at the formal biological definition. In evolutionary biology, a ring species is defined as a connected series of neighboring populations that can interbreed with their relatively closely related neighbors. However, there exist at least two end populations in the series that are too distantly related to interbreed. This phenomenon typically occurs when a single ancestral population expands its range around a large, uninhabitable geographic barrier, such as a mountain, a large lake, or a desert. As the populations expand along two different paths around the barrier, they gradually accumulate distinct genetic mutations. When the two expanding fronts finally meet on the opposite side, they behave as two completely separate species, creating a fascinating biological paradox. Understanding this paradox leads us to why ring species are so incredibly important to science. They provide living, breathing evidence of evolution and speciation happening right before our eyes. Historically, critics of evolution argued that while small changes within a species could occur, one species could never turn into a completely new species. Ring species completely dismantle this argument by showing that small, continuous microevolutionary steps directly add up to a macroevolutionary species-level difference. They also force biologists to rethink how we define a species, since the ability to interbreed is clearly not a simple yes or no question in these cases. Real-world examples serve as natural laboratories for scientists studying biodiversity. To truly understand how these differences accumulate, we can use a simple mathematical model. This formula represents the genetic distance between the original population and a newly migrated population. The symbol D stands for the total genetic distance, which measures how many genetic differences exist between the two groups. The symbol K represents the rate of genetic divergence per kilometer, which depends on natural mutation rates and environmental pressures. Finally, the symbol X represents the geographic distance along the migration route, measured in kilometers. In plain English, this formula tells us that the genetic difference between populations grows steadily as they move further apart geographically. If the geographic distance increases, the genetic distance also increases, meaning the populations become less alike. Now that we have our formula, let us apply it to a real-world example: the Ansatina salamanders in California. The ancestral population started at the north end of the Central Valley, and the migration path down the eastern side is 800 kilometers long. We are given a genetic divergence rate of 0.015 mutations per kilometer. We want to find the total genetic distance. We start by writing down our primary equation. Plugging in our exact values, we multiply the rate by the distance. This calculation gives us a total genetic distance of 12 units. Physically, this means the southernmost population has accumulated a significant amount of genetic variation compared to its northern ancestor. Having calculated the genetic distance, we now need to understand how this distance affects the ability of the animals to mate. We can model the probability of successful interbreeding using an exponential decay function. The symbol p represents the probability of successful reproduction, ranging from 0, which is impossible, to 1, which is guaranteed. The mathematical constant e is the base of the natural logarithm. The symbol c is a sensitivity constant that dictates how strictly genetic differences prevent mating, and d is the genetic distance we just calculated. This equation states that as genetic distance increases, the chance of successful mating drops off exponentially. If the genetic distance becomes very large, the probability approaches 0. Let us continue our Ensatina salamander example using this new probability formula. We want to determine if a genetic distance of 12 is enough to prevent interbreeding. We are given a sensitivity constant of 0.2. We substitute this constant into the exponent, and we also plug in our newly calculated genetic distance of 12. Multiplying these gives us an exponent of -2.4. Evaluating this exponential function gives us a probability of approximately 0.09. Physically, this means there is only a 9% chance of successful mating between the northern and southern salamanders, showing a severe drop in reproductive compatibility. Before we move on to another example, let us mathematically prove why the endpoints of a ring species act like separate species. We start by observing the geographic separation. As the migration distance x becomes very large, our linear model dictates that the genetic distance d must also become very large, assuming a constant mutation rate. This large genetic variation is then fed into our exponential decay model for mating probability. Because the exponent is negative, as d increases, the entire exponent becomes a large negative number. This mathematical setup perfectly mirrors the biological reality of populations drifting apart genetically as they migrate around a barrier. Continuing our derivation, we look at the limit of our probability function. Mathematically, as the genetic distance approaches infinity, the exponential function approaches 0. In our specific salamander case, the probability dropped to just 9%. Because this probability is so incredibly low, the populations are effectively reproductively isolated. They act as separate species. This mathematical result proves the core concept of ring species, that continuous, small, microevolutionary steps directly compound into a macroevolutionary species-level difference. The math confirms exactly what biologists observe in the field. Now that we have proven how isolation happens along one path, let us look at what happens when a population splits and travels down two different paths. This formula calculates the total genetic divergence by adding the divergence from a western path to the divergence from an eastern path. We use the same mutation rate k for both sides of the mountain. However, we must account for the different geographic distances traveled by each group, represented by x west and x east. By calculating the genetic distance for each route separately and adding them together, we find the total genetic gap between the two populations when they finally meet on the other side. Let us apply this two-route formula to the greenish warbler bird, which expanded around the Tibetan Plateau. The western route is 1,500 kilometers, and the eastern route is 2,000 kilometers, with a divergence rate of 0.004. First, we calculate the genetic distance for each path. The western population accumulated 6 units, and the eastern population accumulated 8 units. Adding them together gives a total genetic distance of 14 units. Next, we plug this combined distance and a sensitivity constant of 0.3 into our probability equation. The result is approximately 0.015. Physically, this means there is only a 1.5% chance of them interbreeding. Let us analyze the profound implication of this 1.5% mating probability. We have established that the two evolutionary journeys compounded to create a massive combined genetic distance of 14 units. When the eastern and western warblers finally meet in Siberia, their genetic makeup is so distinct that they simply cannot recognize each other as mates. The extremely low probability confirms reproductive isolation. The geographic ring is physically closed, but the biological species is broken. This completes the ring species phenomenon, proving how two separate evolutionary journeys create a brand new species barrier. To visualize everything we have calculated, let us look at this geographic model of a ring species. In the center, we have a large, uninhabitable geographic barrier, like a mountain. Around it are distinct populations, labeled A through F. Notice the black arrows connecting adjacent populations, like A to B and B to C. These represent continuous gene flow, meaning neighboring groups can successfully interbreed. However, look at the top, where population A meets population F. Instead of an arrow, there is a red X indicating reproductive isolation. Even though genes can flow all the way around the ring through the intermediate populations, the two endpoints have diverged so much that they cannot mate. This diagram perfectly captures the paradox we have been studying. As we wrap up, let us review the key takeaways from our exploration of ring species. First, they are a beautiful demonstration of evolution in continuous motion, showing that the boundary between species is not always a hard line. Second, we saw how neighboring populations can easily interbreed, while the distant endpoints cannot. Third, our mathematical models proved firsthand how small microevolutionary changes accumulate into major macroevolutionary differences. Ultimately, ring species remind us that life on Earth is fluid, adaptable, and constantly changing in response to the geography of our planet. You now have a solid understanding of this incredible evolutionary phenomenon.